Good afternoon. My name is Mark Sawicki. I am the President and CEO of Cryoport Systems, and today I'll be discussing de-risking the regenerative medicine supply chain, managing ISO 21973 through chain of compliance, and our cryosphere. Before I get into talking about ISO 21973, I just wanted to provide a very short background on Cryoport. Cryoport as an organization consists of 4 different companies: Cryoport Systems, which is the legacy entity, Cryogene, CryoPDP, and MVE Biological Solutions. And each of these entities help support the integrated supply chain that supports the transportation, storage, and related IT and equipment-based aspects of cell and gene. As an organization, we currently support 30 different facilities on a global basis throughout the US, Europe, and Asia, which provides us a comprehensive ability to support any clinical commercial distribution for the cell and gene space throughout the world. And now I'll transition into talking a little bit about ISO 21973. So for those of you that are not familiar with 21973, this is a new ISO standard that came out in the summer of 2020, and it is focused on the general requirements for the transportation of cells for therapeutic use. So it's specifically relevant to the advanced therapy space. A little bit of background about ISO 21973. So in essence, it's the first standard to specifically address in detail the requirements for the cell and gene therapy supply chain. And it's focused very heavily on the distribution aspects of cell and gene therapy, or the transportation related elements, inclusive of packaging, storage, and transportation. And so there's 6 areas of specific key considerations that we've looked at as it relates to 21973. These include cell transportation specification, shipping, container, and labeling, operations, organizational, storage, and documentation. Now, we won't cover all of these in the 15-minute time allotted here, but we will look through some of the more critical aspects of this standard. So walking through the ISO standard, there's certain sections that stand out that are evolutionary as it relates to the traditional transportation standards. And the first is under Section 4 best practices. And so Section 4 best practices covers a number of specific areas related to cell therapy transportation, but most importantly includes equipment performance, cleaning, and equipment use history, as well as shipping container qualification, cleaning, and disinfection. So these are some critical areas that haven't traditionally been focused on when it relates to distribution of key pharmaceutical or biopharmaceutical materials, but are critical to the cell and gene space due to the fact that many of the materials that are being moved are critical raw materials like apheresis and leukapheresis samples. And then many times these going to clean room or clinical environments where sterility is of concern. Secondly, is these materials are in many cases irreplaceable, which means that you can't send another batch or another aliquot of the material if there's a failure event that occurs during transportation. And as such, the qualification of the equipment and the sterility of the equipment are really critical to successfully support this space. And so we've looked at a couple of different areas that really focus around those best practices. First and foremost is what we call chain of compliance. And this is providing comprehensive traceability of the equipment and the processes used in managing the commodity itself. So things like equipment performance, requalification, calibration history, in-field exception reporting, as well as requalification and hold time performance of the equipment. And then secondly is the VeriClean processes, which are related to what we do, which is the first validated cleaning and disinfection process for transportation-related equipment associated with cell and gene distribution. Moving into a couple of other areas from a discussion standpoint, best practice under Section 5223. And so this is really focused around storage and handling conditions. And one of the new emerging elements that tie into this is the segregation of human versus animal-derived products to prevent cross-contamination. And this requires traceability standards for the equipment so that you know the historical utilization of that piece of equipment as it relates to the commodity that's been placed within it. And being able to understand that the equipment that is being used to transport cell and gene therapy materials has only ever been used for that exacting purpose. Cryoport has actually addressed this using what we call our advanced therapy shipper line, which are units that are dedicated exclusively to human cell and gene therapy distribution. And those allow us to create full segregation of not only equipment, but the processes and the accessories that are also associated with that distribution. Moving on, the next area of interest that we have looked at is section 5234 of the ISO standard. And this relates to transportation related issues associated with the transportation service provider and trying to eliminate or avoid certain specific occurrences from occurring during transportation. So things like vibration, shock and drop, orientation of the package, anti-tampering, as well as X-ray imaging. And some of these things are avoidable through process controls, but others are unavoidable in the transportation process, regardless of how critical or careful the provider that's supporting this distribution is. And as such, we've had to look at a couple of unique service platforms to address and support that. So the first is what we call our Cryo Shuttle, which is our own physical service fleet that we have trained drivers that understand comprehensively the distribution requirements for these type of packaging. That'll give us much more control and risk around misorientation and physical handling of the packages. The second, and more importantly, is the emergence of what we call our cryosphere. And so the cryosphere itself is an emerging piece of equipment that is an orientation independent cryo shipper, which reduces the handling risks associated with transportation, so that the actual cryosphere can be placed onto aircraft at any orientation, and the payload capacity of the payload compartment will always remain in an upright position, eliminating the risk of a sideways orientation from acceleration of the nitrogen evaporation rate. In addition, it has substantial improvements related to ergonomics, which allow it to be moved much more easily within a clinical or hospital setting with integrated wheels and a handle, as well as the ability to put it onto narrow body aircraft. Secondly is section 622, and this ties into performance, commodity, cleaning, and maintenance records associated with the equipment itself. This goes back to that traceability that we talked about earlier under chain of compliance, and the fact that the criticality of being able to demonstrate all of these records for the entire usage cycle of the piece of equipment. So you can go back and not only look at the most recent usage, but you can go back to every usage over the history of that piece of equipment in the field and understand how it's performed, its calibration records, its usage history, its commodity history, the physical locations in hold time and things like nitrogen evaporation rate. And this will also tie into the cleaning validation as well. And all of these things are supported through an informatics platform, which we call the Cryoportal, which supports and manages all that traceability and maintains every piece of data, not only for the process controls related to that equipment, but also on the transportation itself. All of the data that comes in from the data logger and the transportation providers in perpetuity, which allow us to really strengthen the AI or analytical aspects of the software to manage risk. In addition to that, we've been really focused on next generation packaging to support risk mitigation in the field. And so we talked briefly about the cryosphere, which is the cryogenic distribution of cell and gene therapies, and the fact that this will be coming out over the summer of 2021. But in addition, we've also designed best-in-class packaging that's related to all of the other temperature ranges associated with cell and gene transport, from -80 degrees up through controlled room temperature, and providing much better insulated hold times for any of those packaging ranges, as well as enhanced analytics as it relates to data management and data collection, but also fidelity and reduction of things like standard deviations in the payload carrier, customization of payloads to better control the commodity configuration, and being able to have more customization on things like the pack out associated with extending hold time dramatically over the standard equipment that's in the space. Moving on, talking through traceability. So traceability is covered under section 7.2. And traceability ties back into the chain of compliance, as we had mentioned, and as well as the cryo portal itself. So it's key to control that data, and it's key to be able to access that data similar to what you would see in a GMP manufacturing environment. Or you can go back, and if you have an audit request from a regulatory agency, you can go back and actually pull the data for the entire usage cycle of that piece of equipment in question, so that they have the historical traceability of that particular equipment and the processes associated with managing that equipment. In addition, there's concerns around storage facility under section 9.2. And this ties back into cleaning of equipment and premises to prevent cross-contamination, including frequency and method. And so that we've talked extensively around the equipment-based aspects, but this also ties back into the facilities and supply chain related storage and distribution and fulfillment competencies as well, and making sure that you maintain that cleanliness process, so you're not introducing contaminated equipment into a long-term biostorage facility, for example, or clinical facility, and as well as the facilities within, that are being controlled and supporting those storage of those key commodities in drug product. And so Cryoport has moved in the direction of actually launching its own bioservices competency, which is co-located with our logistics centers. And we call them supply chain centers, which meet all of these process-related controls associated with the best practices around equipment management, and as well as cleanliness and cross-contamination. Finally, talking through section 10, which is another area of key importance, which is documentation. And so one of the areas here that's important is a history of shipping containers utilization should be documented and retained when the shipping container is reused. And so one of the keys here is the ability to have that traceability and control the packaging associated with that distribution. And so if you don't own your own packaging and you don't control the validation and qualification aspects of that packaging, you cannot adhere to the regulations highlighted under section 10, or it's much more difficult to do so through a partner. And that's why, as Cryoport as an entity, we've been very, very focused on ensuring that we directly are able to maintain all of our equipment and all of the processes and all of the data that's associated with that historical qualification and historical process management. So just to summarize, obviously the power of the new ISO 21973 standard is its development of a clear path to optimal efficiency and risk mitigation. And as the first standard in this space for cell and gene that is starting to look at and address the critical considerations associated with managing materials that are irreplaceable and that are coming in and out of, in many cases, controlled environments. And so we think it's a step forward for the industry, and it will provide much better standardization and optimization of processes associated with the supply chain associated with cell and gene therapies. So again, the 6 areas to consider are cell transportation specification, shipping container and labeling, operation, organization, storage, and documentation. I appreciate your time today, and I'm more than happy to answer any questions offline, as this is a virtual event. Thank you very much.